You're listening to Rebecca Maschke from IQPC, and I'm speaking today with Dr. Jörg König from the Institute of Experimental and Clinical Pharmacology and Clinical Toxicology at the Friedrich-Alexander-University in Erlangen-Nürnberg, Germany. Since 2005, Jörg's focus has been on pharmacogenetics of drug transporters and characterization of the functional consequences of polymorphisms in transporter genes. He also specializes in analyzing the role of transport proteins in drug-drug interactions using in vitro cell models and in vitro in vivo extrapolation of transporter data. Good morning, Jörg.
Good morning.
Jörg, which cell systems do you routinely use for the analysis of transporter-mediated drug-drug interactions, and what are the advantages and disadvantages?
Okay, routinely for the analysis of, let me say, we're focusing on uptake transporters normally. For uptake transporter analysis, we routinely used human embryonic kidney, so-called HEK cells, stably expressing the respective transport protein. With these cell systems, we are able to standardize our uptake assays, and we can really compare. For example, regarding drug-drug interactions, we can compare the uptake of the single drug with the uptake of the single drug in the presence of, let me say, an interacting partner. Using these HEK cells, they have really great advantages regarding the standardization of the uptake assay, because when you use always the same, cell density, for example, you can really compare different uptake assays from different days, and you have the possibility to standardize your uptake assay in a really good fashion. The disadvantages of these HEK cells could be, regarding the substrate you're using, the endogenous background of transporters normally present in these cells, and this background can lead to also high uptake rates, for example, with the substrate, and that means that the difference between the transfectants and the control cells you have routinely to use to calculate your net uptake could be very small. The second cell system we are routinely using are MDCK cells. MDCK are maiden Derby canine kidney cells. These cells grow in a polarized fashion. That means when you cultivate them on filter supports, they had so-called basolateral membrane domains, and they had apical membrane domains. And by the use of these cells, you were able to study not only the uptake, but also the vectorial transport of substances and of drugs through the cell monolayer. For this, you need not only single transfected cells, but you need, and that we have established several years ago, so-called double or multiple transfected cells, simultaneously expressing an uptake transporter in the basolateral membrane and an export pump in the apical membrane. Using this cell system, you can add a drug or two drugs to the basolateral membrane and can look at the vectorial transport and the appearance of the drug in the apical membrane, or over the apical membrane, in the apical compartment, so that you can measure not only uptake of one drug, but also the vectorial transport, mimicking a little bit the so-called, for example, hepatobiliary elimination of drugs. The disadvantages of these cell systems are that they also have large background of endogenously synthesized transport proteins, and that they also express export pumps in the basolateral membrane, transporting substances back in your basolateral compartment, and this would give problems with the interpretation of the results, for example, when you add an interacting partner. These are the two cell systems we are routinely using in our laboratory for the analysis of drug-drug interactions, or in the case also for the functional consequences of polymorphisms.